Just want to thank the Cohen Foundation for putting today together and for everything that Ben has done. We'd just like to welcome you to our panel on clinical care and therapies for infection-associated chronic illnesses. I'm so excited to be here today and introduce you to our wonderful panelists. They are doing cutting-edge work and are leaders in their field. They're here to help us understand the innovative research and clinical therapies that they're developing that target infection-associated chronic illnesses such as Lyme and long COVID. I'll introduce our panelists briefly, and each will present, and then we'll have an opportunity for Q&A at the end. So I encourage all your questions. So we'll get right into it to very briefly introduce our panelists. First, we have Mr. Matt Tyndall. Matt is the Executive Chairman and CEO of FlightPath Biosciences. He has extensive leadership and operating experience in life science and healthcare technology companies. And FlightPath's mission is to eradicate infectious disease through the development of drugs that target specific pathogens. Next, we have Dr. Timothy Hasted. Dr. Hasted is a professor of pharmacology and cancer biology and associate professor of pathology at Duke University. Dr. Hasted's lab focuses on developing targeted therapeutics for cancer and infectious disease, including Borrelia and Bartonella. Next, we have Dr. Stephen Deeks. Dr. Deeks is a professor of medicine at the University of California, San Francisco. Dr. Deeks has spent his career focused on HIV and since 2020 has been using his expertise to study the cause and treatment of long COVID. And finally, we have Dr. Miriam Zachary. Dr. Zachary is a physician and associate professor at the Icahn School of Medicine at Mount Sinai. She focuses on rehabilitation and optimizing performance, as well as treating chronic illness at the Cohen Center for Recovery and Complex Illnesses at Mount Sinai. So thank you to all of our panelists for being here today. And Matt, if you would like to get us started and tell us about FlightPath.
Great. Well, good afternoon, everyone. So FlightPath is a life sciences company. It is the only life sciences company, I think, on Earth, correct me if I'm wrong in the audience, developing a new treatment for Lyme disease. We were founded in 2019 really specifically to bring a new drug through the clinical process and hopefully eventually to market. The drug is FP100 or hygromycin A. It is a narrow spectrum antibiotic that should not disrupt the gut microbiome, which, as we all know, is quite important. We've been working for 5 years to push this drug into the clinic. So licensed it in 2020. This is a drug that was founded or discovered by Kim Lewis at Northeastern University. We've got support, received support from the NIH and IAID team. We completed all of our animal IND-enabling toxicology work, 4 different animal species with the drug, took it forward and scaled up manufacturing, completed a Series A, led very thankfully by the Cohen Foundation. And then we initiated a phase 1 clinical study. I think Mayim Levy recently said in one of the previous sessions that microbiome is not something that you necessarily want to make less diverse. And quite frequently, patients with Lyme disease use multiple antibiotics in multiple courses. That gut microbiome has been shown in hundreds of papers to be important in preventing chronic disease. And so we thought it was important to focus on narrow spectrum approaches to this disease. And so the target product profile was essentially kill the bacteria, kill the pathogen without harming the patient. The drug is pretty unique. It is actively transported across the borrelial cell membrane, actively transported into the ribosome, and then is bactericidal. Some of the antibiotics used today in Lyme disease are bacteriostatic. This drug happens to be bactericidal. It kills the bacteria by blocking protein synthesis, so it's potent against the bacteria. What you see on the left-hand panel is Kim Lewis's lab in an Erlenmeyer flask. This is little Streptomyces hygroscopicus bacteria making this product. So it's a fermentation model of production. Bacteria essentially excrete hygromycin A. We collect it, we purify it, and as you can see, those huge vats on the right, this drug has been moved from the lab scale all the way over to clinical and commercial scale. And what you see on the far right is actually Swedish orange capsules that are carrying this drug, 400 milligram capsules, and then on the bottom a vial that is being used in clinical setting. This is the clinic that we're trialing the drug in now. We're conducting a phase 1 clinical study in up to 72 patients. These are human subjects. This is a safety study with 400 milligram allocations of the drug, and those patients are in that room receiving this drug over time. It's actually just a safety study to see if the drug is safe and tolerable by patients. This study started in May. It will complete roughly, we'll have data back in, I think, February, and we'll be able to report on it then. The reason that we do this in Australia is that we get a 43% tax subsidy, non-dilutive cash back from the Australian government, which is very helpful. We started this experiment as a company, and again, we're the only company, I think, developing a new treatment. We started to prove a point, which is that all of the great basic research that's done out there, it doesn't treat a patient, right? We want to drive something into the market that actually can be used by doctors to treat a patient. And so we are advancing this drug as an example and priming the pump, if you will, for other drugs to come through and proving that this drug has market value to the investors that support us. So it's really important that we have a big first win here with this drug so that investors understand the market space, understand Lyme disease and its value, and continue to fund new programs. So, and hopefully once we're done with this, we'll repeat it in other tick-borne illnesses and infections. Just a really quick note. We have received financial support from all foundations, the most important of which is obviously the Cohen Foundation, which was courageous enough to come into a Series A round that had not happened before. And so they're very unique and courageous in their approach to funding a private company. And so thank you very much for that. Tim?
Hello. My name is Tim Hasted, and my foray into Lyme disease came through a close colleague of mine, Neil Spector, who we'd worked together on some cancer projects, and some of the things that we had developed in cancer fascinated him enough to ask if we could do the same thing. And we were working on molecular targeted therapies for cancer, and we put up to 3 compounds, novel compounds, into patients all the way to the clinical trial, both in a spin-out setting and academically. And what's shown on this slide is the spirochete. And what we're trying to do is take the same approaches that we've used in development of those cancer therapies, the same idea, these, we call them molecular targeted therapies. And in this particular project, we're trying to image Lyme infection in people to answer the question I think Monica had put at the top of her list was, particularly in chronically infected patients, do they have an active infection? Could we see that in vivo with not in a non-invasive way, which we PET? And the protein I'm going to talk about today is the structure shown on the middle of the slide called HSP90 or HTPG. HSP90 is the human form and HTP is the bacterial form. And we're targeting that protein. And one of our molecules sticking in that protein is shown in the colored beads. Oh, you can't hear me? Oh, sorry. Sorry. When we think about molecular imaging in cancer, and this is the latest drug for prostate cancer called PMSA11. So the idea is that if you imagine a disseminated infection in an individual, you can see this, the individual here as these little black dots all over his body. If we could see the kind of pointer are metastatic nodes, and there are PET agents that can define that. So could we develop a targeted therapy or drug that you could see Borrelia infections in a human being in this similar manner, and then actually use that as a therapy? This is one of the drug at the bottom is something we put into clinical trials for prostate cancer that was funded by a large DoD grant, and to prove that in this case the human equivalent of the target in the bacteria can be used to diagnose metastatic disease in cancer patients, and indeed we can, and that is also going to be a PET agent. To derive something like this, it doesn't exist in nature, it's not natural, it's not somebody else's drug. we do this from scratch. We develop crystals, molecular structures of the protein targets. We're particularly interested, obviously, in developing something that would target Borrelia and not the human form. So there's a lot of geeky chemistry and structural biology that goes into this. And after Neil asked me 6-7 years ago to do this, we finally pulled off the whole thing. And the molecule on the left is one of the ones that we're advancing that I can show here through a chemistry campaign to take a molecule that targets Borrelia and the human form at that point, and then through iterative chemistry, which is involves a lot of enzyme assays and binding things, and you end up with the graphs on the right where we show that our molecule will target the Borrelia form and not the human equivalents. It will also discriminate out of spirochete forms of those proteins in assays I won't bore you with, but it's kind of fascinating that we can modify this molecule to make either species-specific or broad-spectrum compounds that we can turn into PET agents. How do we turn them into PET agents? Well, we plug in things we had developed in cancer. These are a number of structures that are our clinical leads that we're developing through animal models now for borreliosis. And on the left-hand part is what we call our ligand. That's that molecule that doesn't bind to the human. And then we tether it to something with a molecular tether that's also been used clinically. And then we're using clinical chelate molecules that you can see by PET. And the take-home story is that through the Cohen Foundation, we have our own PET machine now. So we're becoming PET experts. And we can show in a novel assay we've developed that it binds Borrelia terrifically. In fact, these things are so hot with some of these PET agents. They're metal chelates that I have to wait all day before I can put it in my machine. So there's massive uptake, so it's very promising. We actually developed a new assay, and we started to ask about other bacteria. And I think the reason I show this is Pseudomonas and Acinetobacter, I think, was some other study that we looked at. This does actually bind to those species, and we're working on an idea that you might be able to leverage this as a way to get a argument with the FDA. It's really important that we advance this because it could have utility in multi-drug resistant bacteria. We came up with a fancy new assay using our PET machine, which is really cool, and we can look at multi-drug resistance with it. So I think this project is really coming together now, and we think we could apply it to lots of different pathogens. When we look at all the sequences of our target, they vary differently. We could dial these in and out, and there's a whole career. I wish I was 28 again when I started my lab. I could do this forever. But our next steps with the Borrelia one, we have a molecule HS538, which is our lead candidate. We have the 2 backup ones, and we are looking to get those into animals, maybe a primate study. And I won't touch on Bartonella because we're trying to do the same thing with Bartonella, and we have a structure there that we just started. So, and these are the folks involved and funding. Thank you.
Edge of medicine. So I actually spent the first part of my career taking care of people with HIV before there was treatment. This was basically in the early 90s. Anyone ever saw the Dallas Buyer Club sort of know what it was like when we had, all of a sudden a disabling, fatal illness that was affecting millions of people worldwide for which there was absolutely no treatment. And the agenda then was driven by this activist and advocacy community, which basically had never really happened before in medicine. And I was part of that. I was basically a physician activist, and we were trying anything that we could that we thought would make sense. And I see that happening now in long COVID, and I participate in that. I think it's great, and it's healthy, and we're learning. But in the background, during that period of time, billions of dollars were being spent, mainly from NIH, but from foundations like the Cohen Foundation, foundations like that back in the day, and industry, going about trying to figure out a treatment and a cure. We haven't reached cure yet, but we certainly have a treatment in a very rigorous manner. And there's now debate going on at levels of NIH and elsewhere about how we're going to approach figuring out what to do with long COVID and these other long syndromes, right? We're going to continue to try to hit a home run, do big studies, and try to get people better as soon as possible, which is what the community wants. Or are we going to actually sort of bite the bullet and try to basically invest heavily into a biomedical strategy that will require mechanistic work, phase 1 studies, proof of concept studies, combination studies to get there? Now with HIV, it was easy because we just had to basically just treat the virus. But these IACCs are much more complicated. I mean, the pathogens involved, but we have multiple different pathogens and they're resulting in multiple different pathways and multiple different clinical phenotypes. So it's far more complex, and I think it's going to be a lot more challenging to figure this out. But I'm just sort of here just initiating discussion as to where we're going to put our resources going forward over the next few years. And this, again, these are discussions happening at all levels, particularly at the NIH. And I think we really need to be thinking about investing in specific pathways. There's many of them. They're listed here. And we have probes. We have drugs that can intervene and disrupt these. And we can do these types of studies, and we can actually interpret them. The first thing to do, of course, is to figure out in long COVID the role of virus persistence. Polybio and other foundations are supporting, and other groups are supporting this in a very deep, deep way. And this is sort of the low-hanging fruit. We got to figure this out first. But then that's probably not going to be the full answer. And so we have all these other pathways that I do think we need to be investing in, doing mechanistic work in the preclinical studies and the animal models, but really moving as quickly as possible into people in studies in humans, and getting that sort of proof of concept, seeing what makes sense, what doesn't make sense, and then eventually moving into these larger studies, probably combination studies. That's going to take a while. It's going to take a lot of resources. And I do think that there's desire at the federal level to sort of invest in this. What I actually think is missing, we have great advocacy, great community activists, great need, probably a friendly regulatory environment, and some support at NIH, and these foundations that are developing. I think the biggest problem right now is industry. We don't really have a lot of industry engagement. It's refreshing to hear some of the work going on up here on the stage. I'm not sure how to address that, but I'm here to advocate that in addition to doing all the kind of stuff that is being advocated here, this rigorous approach, I think, is going to be the best way to get to a true cure for long COVID, for other long syndromes like it was for HIV.
Hi, everyone. So I want to start off by saying I'm not the expert on this, but I work with all the experts. And what I want to talk about today is how working with those experts as a physician caring for these patients, how to bring what they're doing to the patient. And it is such a privilege to work with them and to benefit from the research that they're doing, the cutting edge research that we're doing at Mount Sinai at the Cohen Center for Recovery from Complex Chronic Illness that we're doing in the rehab department. So I'm going to present some of the rehab therapies that we are providing for our patients and some of the testing that we do. But some of my colleagues here touched on drug research that's being done as well. And there's some drug research being done in our center as well. So just like Dr. Deeks had alluded to or mentioned and spoke on, is there are so many different ways we see long COVID affecting patients, whether it's gut microbiome, immune dysregulation, dysautonomia, or dysfunctional neurological signaling, and also endothelial dysfunction and microclotting, and several other pathways as well. So what we've realized and what these experts have realized is there has to be, the care approaches to complex illness have to be different depending on what we see, the symptoms the patients they're experiencing, and what the testing shows will dictate our therapies. So it's not a one size fits all for all of our long COVID patients. So we're taking steps towards building a comprehensive clinic and pipeline, and it's changing weekly, maybe daily. Because what we're doing is we're testing patients, we're treating patients, we're getting feedback from the patients, which to me I think is the most valuable part of our center, is listening to our patients, having an advisory board made up of patients with complex chronic illness, and we're pivoting with that. We're also pivoting with the cutting edge research that's happening, nationally and also at home in New York. So this pipeline was changed, I couldn't tell you how many times, but it basically is just outlining that we have testing that's done on our patients. We had 2 sets of testing. You could even see here, it says pre-intake 1, clinical intake, and then pre-intake 2, because that used to be before the clinical intake. And we found that we were falling into the trap of over-testing, and some of these tests become burdensome for the patient. So we heard the patients out. They said, some of these tests I didn't benefit from, but they sure did flare it up. The day is long. How could we cut some of this out? So we do some of the testing that we find beneficial. They meet with a provider, and through that intake with the provider, through learning more about their symptoms, we're able to target our testing further. After that, we start with prehab, which a lot of the time involves breath work and very low-level therapies, then targeted rehab. And I'll go through the slides and we talk about what that means, what our rehab program looks like. We then, of course, rely heavily on peer support and mental health services. We have clinical follow-up with our clinical team, and then we pull in our additional consults for allied health services as needed. So we have our post-intake evaluation and post-intake assessments, and here are some of the tests that if we find we need to look at all the which end organs do we think are affected? What do we need to look deeper into? What do we need to be testing to find the pathway to treat in this patient? So you guys could read this as good as I can, but one of the things that we do that has been very helpful in treating our patients is looking at endothelial dysfunction with the endopat, as well as resting metabolic rate and looking at mitochondrial dysfunction, 2 things that we have an idea of how to treat now. Then we have the prehab that I alluded to. So we work with Mayo Health or Stasis to do breath work with our patients that they're able to do from the comfort of their home, which is something we try to do as well. We do a lot of virtual visits for these patients. Their clinical intake is virtual, and try to save them trips and exertion. So through prehab and rehab, we focus on pacing. We focus on hydration for our POTS patients. And again, this is a very, 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 very abridged version of things that we do, trying to cram it all in here. We talk about different things that can help with their symptoms, optimizing their symptoms before we get them into rehab. So here's the breath work. I'm looking at the time, so I'll try to go quickly. We found that there is a lot of benefit from the breath work. And what we counsel our patients on and say is it should feel like work at first. This isn't just meditative. This isn't, it should feel like rehab. First, it should feel like work, and you should be getting better as you go and with consistency. Then we have the rehab, which it's so important to note here that we're not working on deconditioning. We're not telling patients to work through their symptoms, which we've heard so many physicians telling patients before. It's in your head, work through it. You're just deconditioned. That is not what we are working on, and that is not what we believe is going on. And there it is, I got ahead of myself there. So we're not working on aerobic fitness. We're slowly retraining autonomic responses to different positions even. So we start a lot of our rehab supine, as you can see here, and if that's what our patients can tolerate, that's where we stay. And we retrain the autonomic nervous system. Peer support and mental health services, additional allied health services such as OT, speech and language pathology to help with the cognitive rehab, neuropsych, if appropriate, for cognitive rehab as well. We also work with a dietitian that can help our patients, and we're working with different formulary pharmacies in the clinic as well. Here's our long COVID course. And of course, there's a role for medication too, but I knew there were experts on that to my right, and I look forward to taking questions.
Well, thank you, everybody. So we'd like to open it up to questions from the audience. If you don't have, oh, beautiful. Thank you.
First and foremost, thank you for everything you're doing. So a little background on me. My name is Armando Guerra. I'm currently a grad student at University of Michigan getting my MBA. Prior to this, I was in the military for about 7 years. And I quickly went from peak physical fitness to effectively disabled by long COVID. I think my question is, there seems to be very few standards of care. I've seen probably 40 providers across 3 states, 6 hospital systems to include the VA. And my experiences have been mainly either just getting kind of blown off, like it's in your head, you're a vet, you're a consultant, you're probably just anxious, go take a break, to we don't know how to help you, and we're sorry. So I think I'd like to hear what you think is going to happen in terms of what the standard of care is going to look like moving forward, and then a separate question, not really related, would be how do we raise societal awareness where, frankly, it's seen as an issue that needs to be resolved. Thank you.
Dr. Deeks, maybe that one for you. Miriam.
I'm going to, yeah.
Sure. One of my favorite things that our executive director, Dr. Dave Petrino, says about our center is that if we become a destination center for everyone in the country, around the world for long COVID, then we failed. So I think through the research everybody is doing for infection-associated chronic illness, through what we're doing at the center and the testing and finding these objective, having tests, objective biomarkers, objective findings with EndoPAT, RMR, all the testing that we're doing and trialing and all the drug trials that are going on, and proving to the world that it's not in your head and you shouldn't be doing cardio. We want the center to be reproducible. So we want to create these centers that are treating these patients throughout the country, throughout the world, because we are severely disheartened by hearing too much what you just expressed. So I think one of the goals for all of us working in this field should be to raise awareness and to make therapies, to make testing reproducible, and just continue talking about it and offering it as a model to other centers.
Thanks so much for what all of you do. I'm a lifelong patient since childhood, and I exist at the intersection of multiple IACCs. I have long COVID, ME/CFS, chronic Lyme, and others, but keep it short. I've had a somewhat spontaneous remission in the last year, and that's why I'm here today. I fear this remission won't last. For a patient like myself, what would you recommend as the most efficient path to try the latest approaches?
It's a great question. I would love to know the answer to that one.
I think plugging yourself into a center that exists treating these things, because, again, being a physician who is not the expert but working with the experts, it's all cutting edge research. So if a physician is part of these programs, they have access to all the cutting edge research going on, all the therapies that are going on, the clinical trials and the testing.
But I mean, now that you're sort of in a remission, I think the idea, the goal would now be to prevent anything that would precipitate another, which is probably exposure to an infection, right? But you have to live in the real world. I mean, I would have a plan, right? I mean, obviously COVID's something you need to avoid, but it may not happen. So what are you going to do if you're exposed? And so these are the types of things that we talk about in our clinic is, people who've had re-exposures. How aggressively should we treat people? Can we prevent, if you have an exposure, put people on? I mean, these are the kind of things that I think you would talk through with a clinician is how to manage these exposures, because it sounds like you're, for whatever reason, have some genetic background that makes you highly susceptible, that whatever all these different infections are inducing some pathway resulting in some kind of outcome. We probably have no idea what that pathway is, and so the best idea now is to prevent those infections in the first place, which is going to be impossible, but have a plan to manage them if you do get exposed.
But I have a different perspective on this too, and it seems like we all talk to patients all the time that are sort of N of one. They have very unique symptomatology, very unique infectious background. A couple of the other speakers and I were out in the hallway earlier, and it seems that we need an ARPA-H grant to fund a massive biomarker study, and it's just, that is the linchpin. Before we get to individual cases, having a massive study of patients who are N of one, an IACI consortium, if you will, where patients can come in and give their samples, hook their EMR records up for years, and let AI do the trick, because this is a non-human problem. This is not something that an individual researcher is going to figure out. This is something that we need help with. But we need massive numbers of samples and patients to participate, and I think that's, not to get to your specific case, but that's how you solve many of the cases out there that seem to be odysseys.
Yeah, I completely agree with that, because I understand where you're coming from. As much as we want a plan, I think, okay, if I get bit by a tick tomorrow, what do I do? Where do I go? So I would love to know, what's that plan? I get calls from strangers all the time. I don't know what to do. I just got bit. Where do I go? And I think, having a clear biomarkers, clear testing to know, okay, yes, you have it. Now here we can take steps from here.
Oftentimes those grants won't come to an individual research organization. Oftentimes those grants come to companies. There's one company in this space right now. So if you want to talk about it and you want to put a massive grant together, multidisciplinary, multi-institution grant with a company that's at the front, I'd be happy to talk to people about it.
Thank you. Any more questions?
Thank you all for speaking today. I actually have two questions for Dr. Haystud. I found your study on the PET scan very promising, and I was wondering, first question, do you think genetic variation in spirochete and how they can become resistant to antibiotics, do you think that could affect targeting them in your scan? And the second question is, what would you see as the largest barrier to moving your study to human trials?
First question, no. Sorry. No, because the target we're targeting is a non-essential gene. It's high copy. It's not likely to change. It's not under drug pressure, under classical conditions, so no. And the biggest challenge to us right now is money. It's like we are ready to we have about a 6-month runway before we flame out, I reckon. Maybe we'll make it to the end of the year, but to do an IND, which we've done in the past, that we know how much that costs, we've done them. We're going to spend a lot of time trying, instead of being in a lab doing stuff, thinking about that, how to make this a reality, and how to do it. And getting it into people to test the idea, does it actually work? And I think we found these other organisms that the CDC care about, as they do care about Lyme disease actually, but that may be leverage for us, because if it works in those organisms, and we know it'll work in Borrelia, then it's there at least, that people can, just like any antibiotic you could use this PET agent, the challenges in this field are actually the same as in cancer, even though that PMSA 11, to get that, it's any consolation to you guys, to change practice for this something that works, that actually is curative. You'd think they'd be all over it and all men would be getting this, but it takes time to convince the medical community this is different, and it's a process. It's that you have to prove that it works before you take away a treatment that does work. And I'm very cognizant of that, and what we're doing, let's prove that it works before, because the same thing we're developing for PET could be turned into a therapeutic using radioisotopes. that's a big, massive leap to use that to treat an infection. You really have to have a good reason to do it. But in chronic infection where there's nothing else, maybe that's the way to go. But before you do that, you have to be sure that it works. So it's a challenge, but not going to stop. So.
I just have a comment related to Dr. Deek's point about the lack of industry engagement in and around these topics. And I have a company that has been fundraising over the course of the last few months, and I would say 9 out of 10 early stage investors that find out that we're interested in long COVID get turned off by that because of the nascency of this area. And I think that there's an opportunity to bring advocacy groups and foundations in an engagement and conversation, particularly with early stage investors, to coalesce and to develop a strategy to build infrastructure around this important, part of the ecosystem. Thanks.
No, I agree, and there are examples of how particularly foundations have used their funds to sort of de-risk. I guess that's what's happening with your company, with these early investments, but also basically we're even working with big pharma, who's got assets that they're just unwilling to test because they don't see any market, the outcomes are too vague, they're too busy trying to cure cancer where they think all the money is. But with foundation support, you can go up to these companies and take their existing assets and then, do these sort of proof of concept studies. Because I agree, I've been knocking on doors of industry since August of 2020, and I would say, we've 20, 30 legitimate discussions have resulted in basically 2 proof of concept studies that we have going on in the clinic right now. I mean, so that's been our hit. Yeah, and so we need to do things differently.
There are good precedents for this. Cystic Fibrosis Foundation, Multiple Myeloma Foundation, Epidermolysis bullosa. There are multiple predicates for this, examples of this, that where it worked, where a foundation that was scaled enough to make a life sciences type investment came in, made an investment, and then when that drug hit market, repaid the foundation so they could do all the work they need to do to keep going, to advance the research. So there are precedents for it. The question is, are the foundations big enough to support the work that you need to do? Because it is a long and risky road. And you need foundations that are big enough that can actually do the work that you need to do, to Tim's point, to prove the drug works and is safe.
All right. Well, that's all the time we have for it. Thank you, everyone, so much. I just, on a personal note, want to thank all of our panelists because we need you, and I'm so grateful for everything that you guys are doing. So appreciate it, and thank you, Ben. Thank you.